Okay, so welcome to this next video in which we are looking at antithrombotic mechanisms of the endothelium. Okay, so far we've seen two anticoagulative mechanisms. So both this mechanism involving antithrombin-3 and this mechanism involving thrombomodulin involves stopping the coagulation cascades to try and prevent the production of fibrin from fibrinogen, okay, and to stop the deposition of fibrin strands, which is so important for making a meshwork that holds together the platelets. Okay, so now let's have a look at some mechanisms which actually stop the platelets from aggregating. Okay, so the first thing is that on the surface of endothelial cells, so let's say here is our endothelial cell, okay, and here is our basement membrane which the endothelial cell is sitting on here. On the surface of the endothelial cells, you have enzymes which are ADPases, okay, so this enzyme is an ADPase. So it's going to break down adenosine diphosphate, ADP. Now, where have we seen ADP? Well, we saw it in the chain reaction for activating platelets. Okay, and it's going to break ADP down into AMP, adenosine monophosphate, and it's going to have broken the beta phosphate off of ADP. Okay, so let's remind ourselves of where ADP was important. So when those initial platelets got activated by collagen or tissue factor, their response was to start chucking out ADP and thromboxane A2. And the ADP, remember, was responsible for creating a chain reaction where a few platelets becoming activated led to a huge number of platelets becoming activated, because the ADP could bind to the ADP receptor and cause the activation of the next platelet along. So you could get massive activation of platelets via this chain reaction. Now, the endothelial cells have an enzyme for degrading ADP, i.e. for stopping this message from getting to other platelets. So, if the endothelial cells are very effective at breaking down this ADP, what you can do is stop the chain reaction. Basically, you can stop one platelet becoming activated, causing other platelets from becoming activated. And remember, if the platelets don't become activated, then we don't get this massive production of thromboxane A2, which is responsible for the vasoconstriction and also the activation of the glycoprotein 2b/2a protein on the surface of the platelets to make the platelets sticky. So if you don't get the activation process, you're not going to get the release of thromboxane A2, and you're not going to get the platelets becoming sticky, so you're not going to get platelet aggregation. Okay, and that's the role of this ADPase on the surface of the endothelial cells, to try and prevent that chain reaction of platelets becoming activated, and to try and therefore stop the rise in thromboxane A2. Okay, so there's one mechanism. Now let's talk about another mechanism. So we'll now talk about two molecules which are very, very important for stopping platelets from aggregating and also stopping blood vessels from constricting, because remember, the vasoconstriction is also an important part of this. So when you form a thrombosis within a blood vessel, not only does the thrombosis, the thrombus, I should say, rather than thrombosis, when you form a thrombus within a blood vessel, not only does the thrombus stand a good chance of occluding the blood vessel directly, but also the thrombus may will be releasing thromboxane A2, and that will cause the constriction of the blood vessel anyway. So that's going to make matters worse, basically. So, endothelial cells have a number of mechanisms to try and prevent vasoconstriction. Okay, so two things that they release to prevent vasoconstriction. So I'll draw an endothelial cell here. Okay, and here's the basement membrane again. Okay, so they produce nitric oxide and also prostacyclin. Okay, so prostacyclin is often also referred to as prostaglandin I2, and therefore denoted prostaglandin PGI2. Okay, so, but the more common name for this is prostacyclin. Okay, and now they also produce nitric oxide, NO, and this is produced by the enzyme, which I'll draw here, which is called endothelial nitric oxide synthase, or ENOS for short. Okay, but this stands for, endothelial is the E, and then NOS stands for nitric oxide synthase. Now, the more correct name for ENOS is to call it NOS-free, because you find this enzyme in other places in the body other than just endothelial cells. So therefore its more proper name is just to call it NOS-free, so that you don't risk attributing it to only the endothelial cells. Okay, and that just stands for nitric oxide synthase free. Okay, right. So, nitric oxide and prostacyclin are being produced. Now, both of these cause relaxation of the vascular smooth muscle cells, and therefore lead to vasodilatation. So they are trying to keep blood vessels open, basically, nice wide lumens. So vasodilatation. And vasodilatation is the proper word for vasodilation. Okay, some people will say that vasodilation isn't even a word. Okay, so these two agents both cause vasodilatation, and that keeps blood vessel lumens nice and wide. Okay, and they also both have effects to inhibit platelet aggregation and activation. Okay, so prostacyclin stops platelets from being activated. So when platelets are exposed to prostacyclin, so let's say we have a platelet here, then the platelet will not undergo activation, basically. Okay, so the prostacyclin will bind to prostacyclin receptors on the surface of the platelet. And prostacyclin receptors are G protein coupled receptors. So here is our PGI2 receptor on the surface of our platelet. And once the prostacyclin has bound to its PGI2 receptor and triggered downstream pathways, these downstream pathways will stop that platelet from being able to become active. Okay, so let me remind you of what activation of platelets means. So the activation of platelets means collagen or tissue factor triggering a platelet to release ADP and thromboxane A2. That's the initial activation of a few platelets. However, most platelets won't be activated in that way. Instead, they will be activated via the chain reaction, because ADP can bind to the ADP receptor, and that will also trigger activation. So the first platelets will be activated directly by collagen or tissue factor. The others will all be activated by ADP that has been released by already activated platelets. Okay, and then when this one's activated, it will also release ADP and thromboxane A2. So that, strictly speaking, is what is meant by platelet activation. It means the platelet being induced to release ADP and thromboxane A2. Okay, so platelet activation is very necessary in order to produce the platelet aggregate, because thromboxane A2, you need very high levels of thromboxane A2 in order to stimulate the platelets to become sticky. So remember, thromboxane A2 binds to the thromboxane A2 receptor, which then through downstream pathways leads to the activation of the glycoprotein 2B/2A protein, so that it can now bind to a lobe of fibrinogen, and that is how the platelets aggregate together. Okay, so if you can block the activation process, then you stop collagen, tissue factor, or ADP from being able to activate the platelets. Therefore, even if you expose them to collagen, tissue factor, or ADP, they will not be induced to release ADP and thromboxane A2. And if you don't get platelet activation, then thromboxane A2 levels are not going to go up, because the platelets aren't going to be releasing it. So that's going to stop the vasoconstrictive actions of thromboxane A2, and it's also going to stop the thromboxane A2 from being able to activate the GP2B/2A protein on the surface of the platelets, and therefore you're not going to get platelet aggregation. Okay, so that's the role of prostacyclin on platelets. So it stops It stops activation. Okay? So what does nitric oxide do to platelets? Well, it's slightly different. It works slightly later on. So nitric oxide stops platelet aggregation. So it directly stops aggregation. Rather than stopping activation, it stops aggregation. Now, how does it do this? Well, basically, if I get my picture of the process back up again, so here it is. We saw that platelet activation is going to lead to them all releasing thromboxane A2. And then the thromboxane A2 is going to bind to thromboxane A2 receptors, which will then, through downstream pathways, activate the GP2B/2A receptor. And this will then be able to bind to a lobe of fibrinogen, and that will allow platelets to start to aggregate in the way shown here. Okay, so basically what nitric oxide does is it stops this pathway here, this downstream pathway. So when platelets are exposed to nitric oxide, even if the thromboxane A2 levels are really high, then the thromboxane A2 receptor will become active, but it won't be able to trigger the activation of the GP2B/2A protein. And if this protein doesn't become active, then it's pointless, the thromboxane A2 binding to its receptor, because it hasn't made the platelet sticky, because the nitric oxide has blocked the downstream pathway. So basically, nitric oxide stops the platelets becoming sticky, and if they don't become sticky, then they're not going to aggregate together. Okay, so that is how nitric oxide and prostacyclin have antithrombotic effects. Okay, so we'll continue this discussion in the next video when we talk about plasminogen activators.